Thanks for the introduction. So, hello everyone, good afternoon. I'm here to present our work on the unsupervised network discovery for brain imaging data. I'm Zilong, as introduced, from University of California, Davis, and this is a joint work with my advisor, Professor Ian Davison. We work with Lieutenant Peter Walker and Dr. Anna Chifney from the Naval Medical Research Center and collaborate with Professor Fei Wang from Cornell University. So here's the outline of my presentation. First of all, I'm going to introduce fMRI, about what it measures and how it looks like as a fourth-order tensor. In the second part, I'm going to define the problem setting, to discuss how we convert the raw data into a graph before we analyze it, and the outline of the problem network discovery. Then I'll present our method in details. I'm going to highlight our major contributions of using a variation of block modeling and compare with related work. After that, the results from both the synthetic networks and real-world data will be presented to explore the potentials of our method. At the end of my presentation, I'll quickly mention several directions that I think are both interesting and worthwhile for future endeavor. Okay, let's begin. Now let's take a quick look at the fMRI data. So it stands for the functional magnetic resonance imaging, which measures brain activity by detecting changes associated with blood flow. The primary measure is blood oxygen level dependent contrast, also known as the BOLD measure. This is a non-invasive method for monitoring brain activity and has fostered very popular interdisciplinary research between neuroscience and data mining. Regarding its data structure, an fMRI scan is comprised of a sequence of 3D images, and each 3D image is comprised of the so-called voxels. Basically, they're the counterparts of pixels in a 2D image. So hence, overall, an fMRI scan can be seen as a fourth-order tensor, being a sequence of 3D images along a temporal dimension essentially makes fMRI scans spatial temporal data. Although in this paper we focus on the fMRI scans, our method, which we will see it very soon in details, is not limited to brain imaging data. It's applicable for getting insights from a very wide range of application domains, such as astronomy, climate, oceanography, etc., etc. For these data, both the spatial and temporal aspects need to be concerned carefully with their physical properties. So a very important problem with the spatial temporal data is to simplify such multidimensional and high-order data into networks for better interpretability. In this simplified network, the node would be a cluster of cohesive elements in the spatial domain, and they're cohesive in the sense of their temporal patterns, while the edges are the interactions between the nodes, that is the inter- or intra-cluster correlations. For instance, in fMRI, we're looking forward to discovering functional nodes and edges, while in the oceanography data, we want to learn dipoles as well as their associations. After introducing the data, I'm going to define the problem setting of network discovery from the fMRI. So there are several key notes I want to make here. The first one is that our model does not directly take the high-order tensor as input, which means we're not directly decomposing the spatial temporal data tensor. Instead, our method is applied to a correlation graph X. It is a correlation graph converted from the raw spatial tensor data. In X, the vertices are the voxels in the spatial domain, and the links would be the correlations between the voxels based on their temporal patterns. And the second note is that in this paper, we did not apply our method to the entire brain in the experiments, but focused on the 36 slice, which means the spatial temporal data we analyzed before the graph conversion process is a third-order tensor, the xy dimensions in the spatial domain plus a temporal, the time dimension. This can give us very clear visualization of the results and an opportunity to compare with previous work. But please note, our method is not limited to a certain order of tensor, and the model can be applied to the entire brain without changing the formulation. So specifically, the fiber of one voxel along the temporal dimension from the scan tensor records the temporal pattern of that voxel. We calculate the absolute Pearson correlation between each pair of the voxels according to the temporal pattern. Then, after getting this graph X, the node discovery is then defined as simplifying this correlation graph into nodes and edges. The nodes are the clusters of voxels that are coactive, and the edges are the associations between each pair of the nodes. Please note the spatial properties of the nodes must fit the requirements of the application domain. And it's worth emphasizing that a complete network has, as everybody knows, has both nodes and edges, and we're going to discover both in one model. Now that we have outlined what the problem is, I'm going to formulate our method. But before jumping to our contributions, let's quickly review several previous work and related work. So work related to network discovery from fMRI data took different approaches with different focuses. Some of them focused on discovering nodes, and the others can be used to learn edges while assuming the nodes are given. But none of them simultaneously discovered both nodes and edges for a complete network within one model. And in the previous work of our lab, our KDD-10 paper used constrained spectral method to separate the coactive network as the foreground from the background regions which are not acting cohesively from a single graph. The KDD-15 paper then extended this approach to analyze the coactive patterns across multiple groups by learning the unified and contrast cut from stacks of graphs. In the sense of block modeling, both papers can be used to learn 2 blocks from each graph because they used the graph cut methods. So the KDD-13 paper took another direction to discover multiple nodes by using tensor decomposition. It simplified network by using strong supervision. Specifically, it used an atlas of anatomical regions marked with the automated anatomical labeling. This is a guidance to constrain the boundaries of the discovered nodes, and the labeled anatomical regions will serve as components of each node. In this way, the nodes are meaningful in the sense of neuroscience. But this paradigm can be quite limited in practice. For instance, in traumatic brain injuries, aka the TBI, or the Alzheimer and other types of disease, there are different stages of damage where the detailed and precise brain atlas are not always ready for us to use. So in this paper, we achieved complete network discovery with no supervision. Within one model, we simultaneously discovered nodes and edges, but we still guarantee the spatial properties of the discovered nodes to have clinical interpretations. This is how we did it. We formulate the problem as a non-negative symmetric matrix tri-factorization with orthogonality constraint and spatial continuity regularization. This is essentially a variation of the block model. And the input X is a symmetric affinity matrix of correlations, where the F is the cluster indicator matrix. It essentially gives us the nodes. And M is the mixing matrix that records the associations between each pair of clusters. And this is essentially the edges. And the regularization term on F guarantees spatial continuity within each of the learned clusters. So the spatial continuity regularization term is a core part of our method, and its importance can be demonstrated with a baseline model that only conducts block modeling without spatial regularization. Given the strong correlations between nodes from the same functional network, the clustering the voxels purely based on their intercorrelations would result in discovering multiple nodes that are distantly located, as we can see here in the yellow box, in the spatial domain into one single cluster of voxels. In fact, in the resting mode fMRI scan, it is well known that a default mode network exists, as we can see to the left. These voxels do belong to the same network. However, neuroscience application domain demands each node to be a spatially continuous cluster of voxels, since the human brain are formed with anatomical regions, and we want to have anatomical interpretation for each of the discovered node. Moreover, the edges between these continuous nodes are also very important for learning a complete functional network within one single model. One solution that seems to be plausible is to apply the graph cut or two-block modeling approach recursively on the cluster, hoping that the nodes can be separated in the next recursion. However, experimental results showed that instead of dividing the spatially separated nodes, the recursion only kept to trim the complete network to have less and less voxels, while still keeping all the nodes within one cluster. We address this limitation by incorporating the spatial continuity regularization term. It enables us to automatically divide spatially separated nodes while discovering the edges between them. And the theta here is the key of that regularization. It encodes clustering penalty between all pairs of voxels. It is calculated according to the spatial coordinates of the voxels in the form of sort of like a reciprocal Gaussian kernel. The penalty becomes larger as the distance between the voxels increase. Hence, the regularization ensures that if the voxels are spatially far apart, the clustering will get higher penalty to put them into the same cluster, which means the voxels from spatially separated nodes are discouraged to be put into one cluster in spite of their potentially strong correlations. Okay, that's a very succinct introduction to our method. We then present experimental results on both the synthetic networks and real fMRI data. So in synthetic network experiments, the ground truths of the underlying nodes and edges are constructed and are known, and we want to see how our method rediscovered them. From the real fMRI, we answer the question of whether our method is practically useful. And the answer is yes, as we find insights consistent with domain scientists. So in order to create an artificial data that is realistic and captures the key features of real data, we use the real anatomical atlas to help us construct the ground truths. The spatial coordinates of voxels within each underlying node or region is a subset of the anatomical regions from the brain atlas. It is worth emphasizing that we're not using this in our model or solver, but only use this to construct the ground truths. We then generate the underlying edges between nodes and background regions to simulate different types of noisy settings. Overall, we tested 4 kinds of scenarios: the simple network, the degrading network, networks suffering local noise, and networks suffering global noise. We define the local noise as the correlations between voxels within the same background region, and the global noise is defined as the correlations between each pair of voxels where one voxel from that pair belongs to a functional node, while the other from the same pair belongs to a background region. We change the correlations in the yellow circles on this slide with different values to test our method with different degrees of degrading or different levels of noise. Our method only focuses on discovering functional nodes that are the clusters with strong intra-cluster correlations, as well as the edges between them. So on the next slide, I'm going to show 4 discovered nodes for each setting. Everything in the white box here are the edges between the 4 functional nodes, which will be presented and reported on the next slide. So as we can see here, for the exemplar settings presented on the previous slides, our method is able to successfully discover the underlying functional nodes as well as the edges between them. From the numerical evaluations on the node discovery accuracy, we found that the different nodes are not equally well discovered in different settings. One observation is that the node number 4 in the yellow circle tends to be the most difficult to discover. There are several tentative explanations for this. One is that it has very notable irregular shape, and the other is that it is created when it's surrounded by background regions and nodes. This actually further emphasizes the importance of introducing spatial properties to get useful clustering results from spatiotemporal data. So for the real-world fMRI data, we constructed the absolute Pearson correlation matrix X as input and conducted experiments on 3 different cohorts. The control cohorts, which are the young and healthy individuals, the elderly normal cohort, and lastly, the old demented cohort, which are the individuals that suffer Alzheimer's disease. The results, especially the discovered nodes, are compared against each other. Please note, the clinical interpretation of the results are provided by our collaborators, Lieutenant Peter Walker and Dr. Anna Chifney. So we discovered the patterns in the 3 different cohorts that are consistent with neuroscience discoveries. Without using supervision of anatomical structures as guidance, we still guaranteed the continuity of each node. In a representative to the left of this slide, from the young cohort, normal activities are discovered from the parietal and occipital lobes and the cingulum region posterior, as well as the frontal cortex. In a representative in the middle of this slide is from the elderly normal cohort. The parietal and occipital lobes and frontal cortex regions are discovered as seen in the young normal representative to its left, but the region within the black oval in the middle shows some activity on the edge of the cingulum region posterior, but not even as much as the Alzheimer affected patient to its right. This likely indicates normal slowed activity that comes with normal aging. In the representative to the right of this slide is from the demented cohort. The occipital and angular are discovered with retarded activity levels, which are likely due to Alzheimer's disease. Frontal cortex is not discovered as a functional node here in the black oval region. It's completely missing. This shows its hypoactivity and its possible lack of decision-making or executive function. And those are likely due to the Alzheimer's disease as well. And the cingulum region anterior in the lemon region is discovered as a functional node, which should not have been. And this shows the relative overactivity of cingulum that may manifest as depression, and depression is common in Alzheimer's disease affected patients. We also tested our method on each individual of each cohort and present the average network discovery results from the spatial domain. So as we can see here, the key structural properties of the average of each cohort are consistent with their corresponding representatives. So I'm going to take the last several minutes of my presentation to describe several future directions. Firstly, we're interested in extending our work to make new discovery of networks than just from the resting mode or reproducing the default mode network. We're looking forward to exploring the task-driven fMRI scans and discover new networks that are corresponding to various tasks. Moreover, since the spatial continuity is actually not locked to the brain imaging data, but a spatial property that is necessary for mining spatiotemporal data in general, our method can be applied to a wide range of application domains to automatically discover useful simplified networks without using strong supervision. The third direction would be new variations and extensions of our formulation. For instance, extending our formulation to look at stacks of graphs in a RASCO decomposition style model. Another direction would be, it is actually one of our ongoing projects to constrain the mixing matrix M to be binary, and we can put constraints on it to discover more useful insights. My advisor is giving a tutorial on constrained programming, integer linear programming, SAD formulations in Australia right now. And that's all for my talk. Special thanks to NSF grant and the Henry M. Jackson Foundation grant for funding this research. And of course, thank you all so much for listening. I'm willing to take questions.